Everyone. Hope you've all been having a great time and enjoyed your refreshments, are ready to settle in back for the next talk. It's my great pleasure to introduce our next speaker, Dr. Brian Cicali. Dr. Cicali earned his PhD in pharmaceutical sciences from the University of Florida. His diverse professional background includes roles at GlaxoSmithKline, Tabula Rasa Healthcare, and serving as a pre-doctoral fellow at the FDA. He's also a dedicated leader in our community, holding key roles within ASCPT, ISOP, and the International Federation of Pharmaceuticals. His current work sits at the cutting edge of clinical pharmacology, specifically using quantitative methods and real-world evidence to optimize drug safety and efficacy. Please join me in welcome Dr. Brian Cicali.
Well, thank you very much for the kind introduction. Can everyone hear me okay? Great. So yes, before I dive into today's topic, two things I want to caveat. One, unfortunately, I was hoping to have clinical data generated. At this point in time, we are waiting on a final IRB, so I will not get into the verification of my models, but I'll certainly provide the background of the project and how we're approaching this. So briefly, before I dive into the lecture today, I just want to highlight how I fell upon the GLP-1 world. So as you may know, these drugs are huge and now being widely more used as their indications increase. I had previously got into this field looking at GLP-1 interactions with levothyroxine, specifically for GI motility. And then as we got more into that, the question became, well, how do we address these, in a more pharmacokinetic and pharmacodynamic way, because a lot of this is real-world outcomes and retrospective studies. So with that, we started getting interested in, well, how can we answer these questions using models, and then what are the key questions that really need to be answered as soon as possible? And one of the contexts that always continued to come up was GLP-1 receptor agonists, when to use them, when not, in regards to pregnancy. So when do you discontinue them to try to get pregnant? Do you continue them while you're pregnant? And then additionally, when can you reinitiate the therapy? Post-breastfeeding or during lactation and breastfeeding? And so a lot of these questions are key details that need to be answered by clinicians, but a lot of it has unfortunately had to be guesswork to this point in time. So with that, for today's talk, I will give a high-level overview on just what GLP-1 receptors do. I'll talk about GLP-1 receptor agonists briefly and highlight what's currently in the field and market. We'll go into kind of the current knowledge of GLP-1 receptor agonists for both breastfeeding as well as fetal exposure. And then lastly, we'll cover the PK, PD, what's known, what's available, and then go into how we're trying to address this using more physiologically based mechanistic modeling. So with that, GLP-1s and their receptors, so GLP-1 is a peptide hormone that plays a pivotal role in insulin and glucagon homeostasis. Its biological activities are attributed largely to the binding of GLP-1 on GLP-1 receptors, naturally. And one key thing that I think people are really starting to acknowledge more is that GLP-1 receptors are not localized to just the pancreas. They provide a lot of activity in a lot of different parts of the body, which is why the question becomes, how do we successfully use these in developing fetus as well as postnatal development as well? And just as an example here in the plot, you can see, I won't go through each one, but you have a concentration of GLP-1 receptors in the kidneys, the central nervous system, the cardiovascular system, the brain in particular, and then of course the pancreas as well. So with that, GLP-1 agonists endogenously have a very short activity life, about 1 to 2 minutes between them entering the circulatory system and being able to hit GLP-1 receptors and then being denatured due to biological conditions. So the first key was, how do we leverage these type of actions for a longer period of time so we can get the drug to where it needs to go. And there's been a lot of great progress, as you can imagine, on this. And your right side of the slide shows just how some of these additions and modifications have been made. And ultimately, the goal was to extend the half-life of these GLP-1 peptides to ensure that they can circulate throughout the body and get to where they need to go to have the proper activity. And specifically in this case, as mentioned on the slide, it's largely due to the hydrolysis by DPP4. So a lot of these modifications for these synthetic GLP-1 receptor agonists is specifically to avoid degradation by DPP4. And again, there's 7 on the market, largely subcutaneous administrations, but of course there's oral as well. So now diving into more of the high-level PK of how these synthetic receptor agonists work. I apologize if the screen is a bit noisy, but in general, we see a delayed but sustained release over time, which has lent itself to, unfortunately in this case, many times flip-flop kinetics. So to easily pharmacokinetically model these, you have to be aware of the rate-determining step of clearance. In addition to that, they have relatively long half-lives, as I just noted. That's intentional in this case. They are distributed mostly in blood and lymph. So we know that the sites of action, for example, in the CNS and the pancreas and kidneys, we need them to distribute outside of there, but because of their peptide size, they are largely remain distributed in the bloodstream as well in the lymph. They're eliminated mainly by proteolytic degradation, as you could imagine, being small peptides, and this is done mostly in the liver and kidneys. So with that, the question becomes, what's the difference between all 7? If we know that they all have relatively similar receptor affinities, what is the goal? And what is the proof of concept for all these different various purposes? And it really comes down to the way in which it prevents the hydrolytic degradation and therefore selective targeting throughout the body. So when we talk in terms of pharmacodynamics now, these GLP-1 receptor agonists have a lot of different effects. And I apologize if this is not big enough for you to read. I can quickly go through. In the CNS, you have a decrease. I can't even read it on here. You have a decrease in caloric intake, so a lot of mood disorders if you have knockout. So basically, they consider it an appetite suppressant for the CNS. When we talk about the cardiovascular system, we're looking at heart rate changes, as well as decreased inflammation, as well as vasodilation. In the pancreas, of course, we have the insulin and glucagon activities. In the kidneys, we also see changes in sodium excretion. And then in the stomach intestines, we have gastric emptying delaying. In addition to that, we have seen a lot of adverse events, and this is where we'll get to and why we are considering this pivotal to understand in terms of developing populations. So in terms of adverse effects, you predominantly see nausea, vomiting, diarrhea, constipation, and abdominal discomfort that all generally fall under the GI disruptions, and that's just because of the nature of how these drugs work. In terms of systemic adverse events, we do see dizziness and fatigue, and then for subcutaneous formulations, we also see some injection site irritation. So now, all that being said, on the crash course of GLP-1 receptor antagonists and how they work, the question becomes, how much gets exposed to fetus, and what do we do about that? So unfortunately, I'm sure as you all can imagine, the evidence in humans for fetal exposure is extremely limited. We currently have a clinical guidance that says do not use GLP-1 receptors 1 to 2 months prior to getting pregnant and during pregnancy. But of course, this isn't always possible to do. You don't always control what life throws at you, and so there has been some limited data in which tapering of GLP-1 receptor antagonists occurred after pregnancy was initiated, and that's some of the data we have. Largely, the information we have that led to this clinical guidance is from preclinical animal studies, and what they were able to confirm in these studies is that there is teratogenicity in exposures that are equivalent to human dosing. So of course that immediately draws the switch that we should not be exposing developing fetuses to this drug. What the overall observations were was a reduction in weight and growth. Of course, one of the activities and actions of GLP-1 receptor antagonists or agonists. But we also saw skeletal abnormalities, and in particular in mice and rats, we also saw significant changes in gene expression throughout all the target organs. So of course this comes into effect, well then yeah, of course we shouldn't be using these drugs in human populations, especially at-risk populations such as nursing children or developing fetus. However, there's also 2 small observational studies in humans that have been published that show no significant risk in either congenital malformation or even overall pregnancy loss. So the question becomes, are we being overly cautious to the point where we're preventing women from getting proper care through a GLP-1 receptor agonist simply because we want to outweigh the risk? And of course, we have to be cautious, and so that's where we're at right now. But the questions become, is there a safe space in which this can be done without hitting some type of toxic effects that could impact fetal development? And when we switch gears to breast milk exposure, unfortunately, we have the same issue in fetal exposure, that evidence in humans is very limited. And again, our current clinical guidances say do not use GLP-1 receptor agonists while breastfeeding. Preclinical studies in animals have shown that there's about a 2.5 to 50% milk to maternal plasma ratio. And this depends on the animal as well as the particular GLP-1 receptor agonist. And this exposure was then related to also reduced weight and growth in developing pups and gene expression alterations. So all this being said, again, kind of is the same story as the fetal exposure. Well, then we probably shouldn't use these drugs and avoid this type of exposure in developing humans. But again, however, with that big caveat, there are some small-scale studies that use semaglutide in nursing mothers and tried to measure the amount of semaglutide exposure in the breast milk. And what they basically found is that they could not quantify any semaglutide in the breast milk. That being said, their limit of quantification was 5 nanograms per mil. So the question becomes, we couldn't calculate anything more than that, but that doesn't mean no semaglutide was exposed to the child. And then even further than that, if the child is drinking breast milk and it has a small peptide of a GLP-1 receptor agonist, is the GI going to be able to break that down prior to absorbing that? And so again, it goes back to the question of, we don't know that answer, so we better be cautious and not risk it. But there is rationale to say that there may be a safe space in which these can be used for the betterment of the mother without harming the child. But then the question becomes, well, how do we do that when it's very difficult to get these measurements? It's very difficult in this population to do this guesswork, and we certainly don't want to risk any harm to the mother or child. And of course, as I'm sure you all can guess, given the type of conference we're at, the answer is modeling and simulation to try to leverage some of the small-scale data we have to better extrapolate to a safe space of use or lack thereof. And so therefore, the first thing we need to do is get an understanding of what the modeling space currently looks like for these types of drugs. So in this case, we had a small review in which we basically looked through the literature and pulled any type of published model concerning any of the approved and experimental GLP-1 receptor agonists. And what we found is there's essentially 23 pop PK PD models. In terms of PD, we have PD for glycemic control, for weight loss, for adverse events, and this particular GI event prediction, as well as heart rate changes. So there's a pretty good amount of information out there, but again, this is all empirical. There's not much of a mechanism, but at least this information is out there. Then we have some PBPK models. In this case, the PBPK models were all specifically looking at GI motility-based drug-drug interactions. And so what these PBPK models, we call them PBPK models, but all we were really doing is we weren't modeling the administration of the drug, the GLP-1 receptor agonist. We were just changing the physiological parameter of GI motility and seeing if that would impact other drugs, which is very helpful for clinical trial development, not as helpful when you want to try to extrapolate to a special population. Another thing that was used was looking at pediatric exposure matching to try to scale dose if needed. But again, this was a semi-physiologic modeling exercise, and in general, there's no whole body PBPK GLP-1 receptor agonist in the literature currently. And then lastly, we have model-based meta-analysis. So we had another 7 models there. And this was largely for cross-agent comparison. So different manufacturers and different developers comparing various GLP-1 receptor agonists across each other, across studies, and then across trial comparisons. So in the context of all this, and in the context of the question at hand, which is fetal and breast milk exposure, we have some significant knowledge gaps that need to be filled. And so they are specifically, how much GLP-1 receptor agonist crosses the placenta during pregnancy. In other words, can we quantify the exposure that a fetus would be exposed to, and what would be the downstream impact of that exposure? There's also the question of how much of a GLP-1 receptor agonist distributes into breast milk, and then secondarily, how much would be degraded in the baby's GI, and then how much would be exposed to the baby in systemically. And then, of course, the big question is how do we address these knowledge gaps without putting mothers and their developing children in potential harm by doing guesswork? And then again, no surprise here, our hypothesis is that we can use mechanistic modeling, specifically PK/PD modeling, to estimate distribution of GLP-1s throughout the body, as well as into the fetus and through the placenta. So the question is, in doing this, what information do we already have? And is this feasible to even try to implement? So what we have are the physiochemical properties of all the GLP-1 receptors, or receptor agonists. We have system-specific parameters for both an adult mother, a placenta, as well as pediatric models. So all that's there. And we also have GLP-1 receptor agonist plasma exposure data. And as I mentioned earlier, we have PD models for both efficacy and safety measurements in humans. So with all that considered, it seems like this is a perfect opportunity to leverage this type of modeling method to try to start filling in these knowledge gaps of GLP-1 receptor agonist exposure, both in breast milk and fetals. But of course, the needs become pretty heavy. The biggest one is we don't have verified whole body GLP-1 receptor agonist PBPK models. That's fairly easy to address with time and model development. However, how do we verify this? We can show the mathematical framework to do anything we want, but if we're not confident in the prediction is reflective of real-world evidence and real-world exposure, then I certainly wouldn't want to use that for clinical decision-making. So then the needs are, we need GLP-1 receptor agonist exposure measurements in breast milk. and then GLP-1 receptor agonist exposure measurements in the placenta. Both are certainly possible, but again, not the easiest to do. But that's where we started addressing first, was can we partner with some OBs and women who would be willing to do this to provide samples so that we could start quantifying and verifying these models so that we could apply them to a greater population and take some of the clinical guesswork out of these populations who need these drugs, want to use these drugs, particularly with regards to diabetes rather than weight loss, but that still becomes an issue as well. And so how do we best do this in a modeling framework? So the first one would be fetal exposure. And again, we have this large PBPK model schema that's been developed that we are leveraging. And so if I don't know if I have a pointer. No, but okay. So that bottom compartment there. Perfect. So here we have basically the part of the model that's representing the placenta as well as fetus. And then we have your typical whole body PBPK model. And just quickly to highlight, we're not just doing a first order in and out, but we're also doing a semi-mechanistic permeability limited distribution. The goal is to really leverage as much physiochemical property data and in vitro data that we can to try to accurately estimate how much drug is going to pass through these different compartments without having to leverage direct invasive measurement. So that being said, we also have two dosing administrations. So the first one, of course, is the GI. So you can take these drugs orally, but again, largely they're subcutaneous. So we have dosing depots in both the muscle and skin. And that is something that we've previously worked on and are leveraging that from our hormonal contraceptive work to now subcutaneous administration modeling for GLP-1 RAs. The next question then is, well, okay, that's all good, but what about breast milk exposure? So in this case, we've leveraged a somewhat different modeling method. So we're using I guess we could say semi-whole body PBPK model framework for this. The reason for that being we don't want to cause any issues in model identifiability because now we need to model both a young nursing child and a mother simultaneously. So in this case, again, we have our drug depot, so either orally or in the skin. And then we have basically a human breast milk compartment that becomes the dosing site for the pediatric model into the gut. And just quickly, again, the breast milk actual distribution is more semi-mechanistic than just first order in and out. And so again, we're trying to estimate the best we can using physiochemical properties to get to how much drug would be expected to enter the baby's gut. And then once we get to the gut, we have proteolytic and pH changes that we're also modeling and mentoring. And unfortunately, my next slide would have been, this is how the model currently performs, but we don't have that data yet. So I will go right to the challenges and opportunities moving forward, and hopefully in the next few months, I'll have the full verified models that we can start extrapolating with. So again, challenges and opportunity. One of the biggest challenges is data availability for model verification. Because again, what are we willing to assume or extrapolate based on simulation alone? In this type of vulnerable population, I would say not much, because there's very small margin for error. The lack of the whole body PBPK, GLP-1 receptor models that we're currently addressing in our lab. And then even bigger, there's in general this huge need for better understanding GLP-1 receptor agonist use in special populations where clinicians are constantly dosing, but a lot of guesswork and they're not really sure where this is going, how it's impacting gene expression changes both in the heart, kidneys, pancreas, central nervous system, and again, in the context of this talk, especially in pregnancy and lactation scenarios. So the key takeaways here is we still don't have a good understanding of GLP-1 receptor agonist exposure in fetuses and breast milk. And so currently our risk avoidance assumption, so if you can avoid this type of administration during scenarios of pregnancy or breastfeeding, hold. But there is new evidence that's coming out that's suggesting that there may be a safe space for GLP-1 receptor agonist use in the context of pregnancy and breastfeeding. And right now we're in the process of leveraging mechanistic modeling to try to help bridge that gap quantitatively, so we can say if it stays below a certain level, then we are in a safe space. We have no risk to the fetus or developing child. But we're currently on the forefront of getting the data to verify that so we can make that decision without causing harm. And so, with that, 2026 Q4, contact me and I'll be able to show much better information in terms of what that data looks like. But with that, thank you all for your attention. I'm happy to take questions.
Hi, I'm Sarah Coggins. I'm a neonatologist from Children's Hospital Philadelphia, and I've been very interested in this topic. It definitely feels like a class of drugs where the horses left the barn, and it's hard to study in a controlled setting, particularly as people are getting it online or microdosing, et cetera. Kind of clinically as a neonatologist, my concern about these meds from a fetal standpoint is the effect to which they impact fetal growth, particularly like intrauterine growth restriction and the risk for sort of endocrine disruption like hypo or hyperglycemia in the infant. And I'm curious sort of from like a study design standpoint, how to balance a GLP-1 exposed cohort who may be at risk for growth abnormalities or these hypo or hyperglycemic outcomes with control population of infants who may have been exposed to placental dysfunction as like due to obesity, hypertension, gestational diabetes, preeclampsia, et cetera, and like how to kind of winnow out the relative pros and cons of GLP use relative to medication effect versus an untreated mother with complications of obesity.
Yeah, well, thank you. That's certainly a critical issue. The way I've seen it done so far is they basically did the analysis two different ways. The first way was just a GLP-1 exposed, not exposed, and then a GLP-1 exposed versus obesity, preeclampsia, gestational diabetes population to try to tease out statistically if there's a difference between these. Their conclusion was that there was no risk in either, but again, it goes down to these are very short-term studies. So they'll look at the child and say, oh, it's meeting its milestones, but we don't know downstream issues, especially as you're talking about developing your endocrine system, what this will do in the long term. And I think that's where we want to use mechanistic modeling so we can kind of long-term follow these and measure that risk.
Thank you.
Two short questions. So you mentioned about the animal studies. What kind of species did the data come from? Like primates or rodents?
So mice and Sprague-Dawley rats.
Oh, okay. So not even primate data. So the second question is, because it's like more like information I ask for. So since we don't have ability to measure the actual agonist of GLP-1 in fetus and breast milk, is there any synthetic molecule that pretty much prevents that peptide to bind to the receptor, but keeps the PK properties intact, like modifying just one amino acid, this kind of thing. And that would allow us to pretty much track for such molecule PK, but then it would have those adverse effects, and that would be a solution. Is there any way going in that direction from what you know?
Great. I mean, that's a great question. I haven't thought of it like that. The first initial thought that comes to mind is agonizing the GLP-1 receptors is critical for development purposes, as well as various homeostatic properties. So we wouldn't want to necessarily inhibit the binding to GLP-1 receptors to measure the drug with
just PK. You don't even know how much the molecule gets inside. So if you can just make sure it doesn't harm, and then still we know how much of it gets there, and that would be a huge step forward.
I fully agree. As to finding, mothers that would allow us to do that during, gestational growth and breastfeeding. I don't know. If you find one, let me know, and I'd be, yeah.
So I have, if you don't mind, two questions. One is more speculative, but the first question is there likely to be a difference between the orally available drugs now and those that are not, that are given parenterally? That would seem to suggest a difference in stability at the very least. And I wondered if you'd have to develop two different models for that. The more speculative thing is that there's a fair amount of data now coming out about lipids in the liver and the effects on the cardiovascular system and kidneys and whatnot, and inflammation, which would seem to be related, this is the more speculative part, to things like preeclampsia and eclampsia. I wonder if anybody has evaluated that, and it seems to me that the GLPs that are now there are able to reduce that liver profile, and there may be an advantage to treatment if the data on the developing fetus is occurring, well, let me put it this way. The first 3 months is where you're having organogenesis, and so that's a critical time point. So we need some sort of clarification on the exposure that have taken place as to what stage of pregnancy we're talking about. Can you elucidate things for me?
I'll do my best. So your first question, so yes, there is a difference. In terms of model input, it's not too much of a difference because, I mean, for the orals, you really have to ramp up your formulation to get it through the GI without getting catabolized. But of course, that input parameter is going to be different than your sub-Q because your clearance is going to be impacted in different ways. So the amount of drug in your sub-Q that's getting exposed is based on the release, and therefore your clearance is release determined. Your predominant factor is drug release from the subcutaneous depot, not the actual clearance of the drug. Whereas orally, you kind of see that flip back more towards the drug appearing in the system. For your second question, I mean, that's hitting the nail on the head exactly where we want to take this, is ideally being able to show, because these drugs, as you alluded to, certainly seem promising for helping preeclampsia, but no one, including myself, was willing to do that trial until we know it's absolutely safe. I would expect that we will see hopefully in the long term, if these drugs, we are able to determine that the threshold of exposure is low enough, that we'd want to use these later on in pregnancy, later in the 30s and late 20s gestational age weeks, simply because of the organogenesis and all the different ontology that's going on in the earlier stages where, modulating endocrine systems would be very much more risky, for lack of a better term.
Thank you, Brian. I know from my clinical colleagues, this is a really important area and one where we are looking for more information on the use, especially as more young women are taking GLP-1s prior to conception. We had a couple of online questions. I think you've addressed the first one about how you separate out the effects of disease versus the treatment in the control group from our first question here. But the second one was about if there's pregnancy or lactation registry data available that could help inform your models.
I would love that. I mean, I think so. Irrelevant than this, but at ASCPT, I gave a talk in looking at real-world evidence for lactation safety and exposure. And one of the biggest issues is that data is very rarely captured. So it's one of those things, it's a gold mine of data if we have it, but it's not always available. I think we'll see lactation banks become more prevalent over the next few years as the drug use becomes more prevalent. But right now it's very much ad hoc and we don't have a lot of good database sources for this information. All right, well, thank you all for your time.